Hi, in this video series, we are going to solve the DPP of PW Shodh 2.0 batch. This is DPP 2 and from Inheritance Biology, that is unit number 8. Already we have covered the DPP 1, so if you didn't watch that video, please visit the channel. So the first question, when F1 generation of monohybrid cross is allowed to self-fertilize, the expected phenotypic ratio, and we know the monohybrid cross phenotypic ratio is 3:1. Mendel's monohybrid ratio is is outcome of independent assortment of genes. No, this is the this law independent assortment is found after the dihybrid cross. Next, Hardy-Weinberg, no, uncontrolled mating, no. It is actually the separation of homologous chromosome during meiosis. Actually, separation of homologous chromosome. And after the monohybrid cross, Mendel's Mendel's given the law of segregation. Law of segregation. So the next question is capital P for purple is dominant and small p for red is recessive, and similarly capital L for white is dominant and small l for narrow leaf is recessive. When a cross between capital P, capital L, small l cross scheme capital P, capital L, small l in 250 plant, number of red plant and wide leaf. Okay, so red plant, if it is red plant, it will be small p, and if it is white leaf, then it could be capital L dash. This kind of genomic configuration will we have to find out. So if this capital P, small p, capital L, small l is selfing is fertilized with the same type of genome, we can get capital P dash capital L dash will be 9, capital P dash small l will be 3, capital small p capital L dash will be 3, and small l will be 1. And these three we have to find out of 250. So this is 3 by 16. Everything we can say 9 by 16 and 3 by 16 and 1 by 16. So this is 3 by 16 into 250. We can get 46.8, and this is around of 50, and here is the answer 50. Okay, cool. Cool. So let's move to the next question. When a dominant purple color flower, a dominant purple color flower, capital P, are crossed with recessive white, small p, when a progeny will have the purple is to white is. So in that case, it is completely dominant, and this is completely recessive. So uh, we will get all purple. So capital P cross small p, all the progenies will be capital P, small p, and this is all purple. Cool. Next question. Capital RH positive, RH positive, RH positive, marriage with RH negative. Their progenies will be, so RH positive could be RH positive, or this also could be RH positive, RH negative. These two kind of genotype could be possible for capital RH positive male, and if it is RH negative, then it could be only RH negative Rh negative. So in that case, all the progeny will be Rh positive phenotype. But in that case, we could be found Rh positive phenotype as well as Rh negative phenotype. So can be Rh positive or negative. Option 3 is the correct answer. Next question. Question number 6. Typical Mendelian dihybrid ratio for F2 generation. 9 is to 3 is to 3 is to 1. All of Mendelian allele for different trait showed all of Mendelian allele for different trait showed dominance and recessiveness. Mendel only stated about the dominance and recessiveness. Mendel doesn't know about epistasis, doesn't know about codominance, doesn't know about incomplete dominance. In test cross, F1 progeny is crossed with in test cross, F1 progeny is crossed with recessive parent. In test cross, we see some any F1 progeny like that could be capital T, small t, that could be anything, that could be exactly anything. So we can say this is a dash cross small t, or we can say small a small a. This is the test cross. Next, possible combination of gamete which can be found in this from this. Okay, so we know the number of gamete, number of types of gamete, or types of gamete. We say, let's say types of gamete is there is a formula for this, that is 2 to the power n, and n is number of heterozygous. So let's see, let's count the heterozygous. This is this capital A small a is heterozygous, capital B small b another heterozygous, capital C small c another heterozygous, capital D small d, capital E, capital H small f, capital D small d. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. There is seven pair of heterozygous allele are there. So that is 2 to the power 7, and 2 to the power 7 is, let's simplify it. So we can say 2 to the power 2, 2 to the power 2 into 2, sorry, 2 to the power 2 into 2. 1, 2, 3, 4, 5, 6, 7. So 4 into 4 into 4 into 2, that is 16 into 8. Let's say 16 into 4. 16 into 4 into 2. 16 into 4 means 16 into 4 means into 2. 128. 128. Option 4 is correct answer. Please try to write all the calculation you are doing in your mind, so that you can check it is absolutely correct or not. If you do everything in your mind, it is, there is a chance to get wrong. So let's have another question of number of genotype, number of gamete formation from this genotype. So let's count again. This is homozygous, homozygous, homozygous. That couldn't be counted. So capital D small d is heterozygous, capital D small e is heterozygous. So 2 to the power 2 and 4. Sorry, yeah. This will be the on the answer will be 4, 2 to the power 2, but this answer is not given here. So this is incorrect. Question is incorrect. Let's check the solution here. You can see here there has given option 3, and let's check the solution. Oh, there is no solution. But I think there is capital A, capital A, capital B, capital B, and capital C, small capital D, capital C. This from these three genotype, any one should be heterozygous, capital C, small c, or capital B, small b, that kind of thing should be there. And then that answer will, we can say the answer is 8. But if only two heterozygous are there, you can produce only four types of gamete, right? So that's it for today. Thank you. Get such